So today we have Dr. Matthew Nelson, Chief Executive Officer of Genscience, a tech-focused company to improve integration of genetic evidence into drug discovery. Genscience is an affiliate of Deerfield, which Dr. Nelson joined as VP of Genetics and Genomics in 2019. Prior to Deerfield, Dr. Nelson spent almost 15 years at GlaxoSmithKline and was most recently the head of genetics. Prior to GlaxoSmithKline, Dr. Nelson was the director of biostatistics at Sequenom and director of genomics at Esperion Therapeutics. He's a co-author of over 80 publications. Dr. Nelson was an adjunct associate professor of biostatistics at the University of North Carolina from 2010 to 2016, and he holds a PhD in human genetics and an MA in statistics from the University of Michigan. Dr. Nelson.
Great, thank you. And I guess, parenthetically just reinstated my adjunct position at UNC in pharmacology. So we just did a series of online courses around genetics and drug discovery and development. So thank you. It's a pleasure to be here. I think we agreed beforehand, if you have questions as I go along, please feel free to interrupt me and ask those questions. My talk is fairly modular. So I do not have to get through all 48 slides that are part of the main deck. So I'm going to essentially give you a quick background on kind of my context in Genscience and Deerfield. Then I'm going to talk you through kind of how I think about the challenge that we face in drug discovery and development and how we can frame that from a probabilistic perspective in terms of where the challenge in R&D productivity is and how we can influence that with genetics. I'll talk you through kind of some of the main results from our paper that we published earlier this year, which was a revisiting of our 2015 paper on the differentiation of success in drug programs with and without genetic evidence. Then I've got a little bit of an interlude where I'm going to talk a little bit about how I think about where the field of human genetics is today and where I think it's going to be going. And kind of how I feel like we're probably going into, again, a bit of a quiescent period of really key novel discovery in human genetics before it explodes again. And then I'll wrap up with a preprint, if we have time, kind of casting genetics from a drug safety perspective. So first of all, I joined Deerfield. So I left GSK. Everybody knows what GSK is, big pharma company. In 2019 to join Deerfield. It's a firm, a healthcare investment firm here in New York City. We focus on all things healthcare. So it's anywhere from therapeutics to devices, medical technologies, and healthcare services. We manage about $15 billion in assets, and about a third of those we invest in the public markets, and 2/3 of those are in private funds. So we can get involved in anything from new co-creation, seed funding, Series A, B, C, that's kind of early stage to later stage private investment, all the way through, as I said, public investment. So I joined Deerfield from GSK to bring a genetics lens to some of the investment activity. And you'll see kind of why in my subsequent slides. Since I joined Deerfield, I knew that I wanted to have the types of genetics capabilities that I built up at GSK to support Deerfield's investment work, but recognized that we weren't going to be able to build that type of a group inside of an investment firm. So last year we decided to spin the genetics capability out into its own company, Genscience, which allows us to bring on other types of clients besides Deerfield, but that we can use the revenue from those clients to reinvest and to grow the platform and to grow the group so that we can essentially build out the full stack, fledged capabilities in genetics as I see that's value in drug discovery and development. So that's Deerfield, that's Genscience, and these are some of the other affiliated organizations with Deerfield that I won't go into here. You guys are very familiar with Moore's Law. It's amazing that an observation that was made over a few years of data back in 1964 has more or less borne out one way or another to the increasing compute capacity that we have seen over the past 60 some odd years, which is pretty amazing. And that's been enabled by just breakthrough after breakthrough in material science and all kinds of technologies. In drug development, we have something that has been coined Eroom's law, obviously Moore's law in reverse. This this is a figure that was where this clever term was postulated in about 2014, 2012, actually, by Jack Scannell, where they showed that the R&D productivity, that is the number of drugs approved for every $1 billion, or every $1 million here, every $1 million of R&D spend. And you can see that that has gone down some over roughly the same period of time as Moore's law, from in the 50s, roughly 30 drugs approved for per $1 million spent, down to less than one drug. So it's per $1 million spent. And there was actually an article in The Economist earlier this year where they updated the data from this, and we're talking about this Ebrams law. And we actually are seeing, maybe we've kind of hit the plateau, that we're not continuing down in that downward slope, but maybe we've at least flattened out. And there are many possible reasons for this, including increasing use of more targeted therapies, more therapies developed for orphan indications, rare diseases that have a higher probability of success. But overall, the challenge is it is extremely expensive to get a single drug to market. So I want to talk a little bit about why that is. There are essentially three factors that drive the cost of R&D. You've got each of the different phases that it takes to come up with what drug you're going to develop, find the drug, make sure the drug can go into clinical development, and then you have the clinical development cycle, phase 1, 2, and 3. So you have the cost of each of those stages, you have the probability of success of each of those stages, and now it's not trivial to think about the cost of money as well, the cost of capital, based on the increased interest rates. So let's start with the probability of success. These are more or less the accepted estimates for your phase transition to go from target discovery, target hit to lead optimization, preclinical, phase 1, 2, 3, and then ultimately getting a submission to a launch. So in the preclinical stages, so selecting your target, is very, you turn over those projects very quickly with a moderate probability of success. But once you start trying to find a molecule, those probabilities of success individually are relatively high, somewhere in the 70 to 80 percentile range. So then once you get into clinical development, phase 1 has an average probability of success of somewhere around 85%. But then phase 2 plummets. You're at about 30% probability of success in phase 2, somewhere around 55, 60% in phase 3. And then once you successfully complete a phase 3, the likelihood of approval is quite high. But if you individually, they don't look too bad. But if you ask from any one of these stages, what is the probability that I'm going to get to an approved drug? That's what's shown here in red. So under this model, the probability of going from target discovery to an approved drug is about 1%. And even going into phase 1, your probability of getting to an approved drug is only about 14%. And I actually built this model using a fairly favorable scenario that is non-oncology, because oncology's probability of success is about half that of non-oncology, and antibodies. Antibodies, their clinical probability of success is about 50% higher than overall than it is for small molecules. So if this was oncology small molecule, this would be about 3% going from phase 1 to launch, just for comparison. So probability of success is one factor. The other is the cost per phase. And these are roughly, if you kind of average across different types of therapy areas and different modalities, this is more or less what it costs for each one of those stages to get a program from one stage to the next. So if you just sum those up from left to right and asked, once if I do a postmortem on one successfully launched drug, and I ask how much R&D did I spend to get that one drug from an idea to an approved drug, that average over all therapy areas is about $350 million. So reasonable, I guess. But once you take into effect the attrition rate on the left, that's how you get to these incredible estimates of the cost of getting a drug to market. So once you factor in the failures you're at about $1.2 billion. Again, for very favorable circumstances of antibody, non-oncology. This is essentially Regeneron strategy, is focusing on that subset. I use that example because if I do small molecule, it's like $2.5 billion, and the numbers just kind of overwhelm readers. So this is essentially why drug R&D is so expensive. It's not that the individual phases are so outlandish, it's just your probability of success and all of the attrition, the number of programs that it takes to get one successful drug to launch. Now, 70% of the cost and 60% of the time is spent in clinical development. Yet, as I said, there's only about a 14% probability of success of going from phase 1 to launch once you're there. And there's been a lot of analysis of why do things fail in clinical development, especially in the expensive phase 2 and phase 3. Most of those failures are due to lack of efficacy. So roughly 50 to 60% in phase 2 and 60 to 70% in phase 3 fail because the drug just doesn't do what we thought it was going to do when we started the endeavor and took it into clinical development. The next most common reason for failure is safety, particularly in phase 3. Interestingly enough, strategic is really the second most common reason why a project is abandoned at phase 2. There's just, we lose strategic interest. We no longer want to pursue it for whatever reason. The landscape has changed. So anything that we can do to improve that probability of success could have pretty dramatic effects on overall R&D productivity. And we published this paper in 2015 where we had examined, because we've been talking about since the 1990s, the days of the Human Genome Project, once we have genetic information, that genetic information is going to really improve how we select and prioritize targets and develop drugs. But coming into the 2010s, in the era where we're now starting to see lots and lots of genetic data start to emerge, we didn't have any practical empirical evidence that was actually true. We had very few examples where a piece of genetic insight actually led to selecting a target and getting a drug to market. We just didn't have enough time to have those examples at that point in time. And so we did a retrospective analysis where we lined up the drugs, mechanisms, or target indication combinations that had gone into clinical development and those that had overlapping genetic evidence, and asked, did those that have that overlapping genetic evidence, were they more likely to proceed to further in clinical development than those without it? And this was really the money figure when we came up with that, and showed that the further advanced a target indication went in clinical development, the more likely it was to have genetic evidence supporting it. And at the time we published this in 2015, from roughly 2014 data, about 8% of approved target indications had this supporting genetic evidence. And Yufeng was a co-author on that. He and Paula McLean had done some nice work on using linkage disequilibrium with EQTLs. And we essentially use that to help in our variant to gene mapping in a very early application of that in the paper. So we ultimately came up from that paper with an estimate that drugs that were supported by genetic evidence were twice as likely to be successful in the clinic. In the first several years after that paper was published, there was a lot of pushback in pharma around the value of the types of mechanisms that genetic evidence could identify, and whether they were the kinds of programs that they would want to work on. So the next couple of slides, I think, provides a nice practical example of the value of genetic evidence. So in this slide, these are the drug mechanisms for the most prescribed drugs in the United States. And this is from 2019 when I pulled this together. And so you can see that these are all generic drugs that are supported. So you've got the statins as the most prescribed drugs at the top, and then on down. And I asked which of these had similar supporting genetic evidence, as we demonstrated in that paper. And in this case, 9 out of 17, or 53%, of the most prescribed drugs, including the top 4 mechanisms, were supported by genetic evidence, which is, I showed you that 8% rate in the previous slide. The base rate now is probably closer to 15 to 20%. So this is at least, even amongst genetic evidence supporting launch drugs, you've got 2 to 3-fold enrichment of genetic evidence amongst the most prescribed drugs. So genetics is identifying the mechanisms that really are impactful. And what you really care about, if you're a biotech or a pharma, is, am I going to be able to make my money back if I develop a drug? And so I looked at the top grossing drug mechanisms. And after removing oncology and infectious disease, we're left with these 8 or 7 unique mechanisms remaining. And this is the sales in billions on the right-hand side there for these. So the anti-TNFs, kind of dominating the market at that point in time. And 6 out of 7 of these are supported by genetic evidence. So, I think there's pretty compelling evidence that the most impactful drug mechanisms, from their overall impact on human health, as well as the most profitable drugs that are out there, are supported by genetic evidence. So this next section, I'm just going to talk you through some of the key results from our paper. So we followed up that 2015 paper with another paper that was published in Nature earlier this year, where we went back and, we have now almost a decade's worth of new genetic evidence that has emerged from when we first published that paper. We have much better methods now of guessing at what the most likely causal gene is for any genetic association. And there were a lot of questions that were raised in our original paper that we just didn't have answers to, that we tried to address in this paper. And this is Eric Menical, the first author on this from the Broad Institute that collaborated with me on this work. So again, to do this, you need 2 pieces of information. You need gene disease information from your genetics. What is your genetic evidence? And you need target indication information from your drug development activity. So from the genetic side, we use both rare and common, as well as somatic genetic evidence for some of the analyses. So we used OMIM, Open Targets Genetics for GWAS, and then Piccolo is the colocalization paper that we used some of the colocalization results from. GeneBase is the database of the loss of function analysis from UK Biobank. And then Intigen is a somatic driver gene database for cancer drugs. So those are where we kind of pulled together a total of roughly 80,000 unique gene trait combinations for our genetic evidence side of the equation. Pharma projects, the same resource we used in the 2015 paper, is where we got our target indication or drug information. So once we kind of boiled all of that down, we had about 2,100 unique target indication pairs in the analysis. So 809 total targets, 397 total unique indications that went into this analysis.
How many unique traits?
Unique traits. So do we have that? I don't have the number of unique traits, but ultimately it's going to be related to 397. So we used a trait similarity. So if you read the paper, we didn't require that we have a genetic association, say, for hypertension. A genetic association for systolic blood pressure would be good enough for hypertension. So we use this concept of genetic similarity or trait similarity when mapping the traits that were associated on the genetic side of things with our indications. So I don't actually have in this table, and I don't have in my head, the number of unique traits. Coming into it, they're from open target genetics, 49,000 unique traits. But a lot of those traits are very similar to one another. You might have 100 different traits that are around body fat.
And many TDAS studies are actually focusing on a small subset of those.
Exactly. Yeah. Yeah. So, yeah, the study trait couples 57,000. So that's the total number of studies plus with unique traits. So here's the top line from the paper. So relative success is the same metric that we used in the last paper. That's the probability of success given genetic evidence divided by the probability of going from phase 1 to launch without genetic evidence. So that's RS, or relative success. And if we just look at all of the germline data, so this would be the most comparable metric to our original paper, that estimate came out as 2.6. So roughly a 30% increase in the relative success between our original paper and the new paper. And some of that's going to be attributed to just the new genetic information that's emerged, and some of that could be attributed to better methods for mapping causal genes. Mendelian still stands out as more highly differentiated. This is going to be skewed by the development of drugs for Mendelian conditions, which have generally a higher probability of success anyway. And more of those are going to be targeted around the causal gene. But I'll tease that out in just a moment. And then we break it down into other different types of sources. So here's all GWAS, which is a little about 2.2. All OTG is a little bit higher than that. And you can see some of the other sources. The UK Biobank was a bit higher. That's largely because a lot of the evidence is coming around cardiometabolic types of traits like LDL cholesterol, these types of quantitative things that are in UK Biobank analysis that we use. FinGen, Piccolo, GeneBase, they all are roughly around that 2 to 2.5 individually. I did want to show, this is from a supplementary figure. One, so we broke down genetic evidence in lots of different ways to try and tease out, because what we're really trying to do is enable people who are using this information in drug discovery and development or investment decision-making around programs like that. Is there particular evidence that you would weight more than other evidence? And one thing that we specifically teased out was OMIM versus non-OMIM genetic evidence. And so if you look at all OMIM, you've got this highest estimate here, OMIM without GWAS. So sometimes you've got both OMIM and GWAS evidence that overlaps for your indication. GWAS alone, GWAS without OMIM, that's the lowest. That still is about 2. And then the OMIM plus GWAS, that should correspond to similar to Alderline. So there's a little bit different estimate of what's in that. The estimate overall is about the same. So what that shows you is that GWAS and OMIM together is actually a little bit more impactful than either of them alone. Probably relates to the types of traits that could have both complex trait and Mendelian evidence. Things like PCSK9 would fit that bill. Early on in the paper, we show this is essentially looking at your confidence of mapping your causal gene to your genetic association. We used open target genetics for this, and they have this L2G score, and they have as part of the L2G score from their machine learning model roughly a quantitative metric that tells you what is the proportion of evidence or your probability that it's any one of the genes within a locus. And so we used a value of 0.5, meaning that we only at most you could have 2 genes for one association if they were both exactly 0.5. But in practice, for most loci, we're only selecting the most probable gene for any genetic association. And we show that as that confidence increases, the relative success increased as well, suggesting that it is a valid metric. Another question that was raised, right after the original 2015 paper was whether genetic effect size mattered at all. Are genetic associations of larger effect more interesting, more important, more informative from a target selection drug discovery perspective than those with a smaller effect size? And so we tried to tease that apart in lots of different ways. The first thing we looked at was year of first association. So we looked at all of the genetic associations from all the different sources, and we identified where it first appeared in a study, and then did the relative success analysis with that. And we really find that there's certainly no significant difference amongst them. The slightly higher estimate earlier on is related to the fact that you have things like cardiovascular disease and LDL cholesterol in kind of biasing a lot of those early GWAS studies, but that effect is not significant. Second, we looked at kind of polygenicity as a metric. So this is the total number of genes, genetic associations of genes that can be mapped for a particular indication. And so one, which would be a billion, 1,000 would be getting into things like blood pressure, where you have so many of those. And we entirely expected the result of that analysis to be the opposite of what we saw. We were expecting to kind of see an inverse relationship. That is, the more genetic associations you had, the less likely that relative success would get kind of swamped out and would come down. But in fact, we see the opposite of that. And putting this into a linear model, it's the only one of these, all of these different metrics that we look at in this slide that had a statistically significant trend.
But the drug is targeting one of the 1,000?
It's one of the 1,000, yes.
And
So it's not necessarily, so it's not getting at the positive predictive value question, but it is saying that, if you have genetic evidence from, these 1,000 associations for type 2 diabetes, that those mechanisms that had at least one of those supporting evidences was more successful than those that didn't have that. Good example of that is GLP-1R didn't pop out as a genetic association from a GWAS until the one of the more recent papers.
And also depends on how large was the zero study, right?
Yeah, it's how well powered is the study essentially. So it's not really. So it's going to be more related to the power of the study than whether it's a polygenic or oligogenic genetic trait. So it's essentially just saying that the more genetics discoveries you have around an indication space, the more likely the successful drugs are going to have that genetic evidence.
Yeah, so maybe it's a simple question, but when we have genetic evidence, what helps the most to increase the success of drug discovery or drug
Given that you have genetic evidence, I'll actually get into that in subsequent slides. Yeah. But that's, that is kind of the next important question to answer is, so given that we want to work on programs that have some form of genetic evidence, now of the 1,000, how do you pick which one you're going to work on? I won't entirely answer that question, but I'll suggest some ways that we can look at it. And then getting specifically at genetic effect size. So, these things are all kind of correlated with each other to some degree, but we looked at categorical quantitative traits with beta, binary traits with odds ratios, and then kind of as a surrogate for those minor or low frequency. Obviously, you're going to find the more common one first, but they're going to have smaller effect sizes in general. And the bottom line is that we really don't see any significant trend in any of those. So we don't find that effect size has any association with that. There was a paper by 23andMe, it's a preprint that came out after this, that kind of was a follow-on to our study using the 23andMe genetic evidence. And they do find some evidence that effect size could be associated. But I think there's some confounding there too that I've discussed with them. This is my favorite figure from the paper, and one that I come back to over and over again. So here what we've done is we've now stratified the indication space by therapy area. So this is everything together, and then hematology, metabolic, and so forth. This is the phase 1 launch metric that we've been looking at. So this is that 2.6 that we saw from the earlier slide for all. But now here's how it breaks down in different therapy areas. But then we did more. We kind of decomposed that into, well, what is its effect on relative success from going from preclinical into the clinic, phase 1, 2, 2 to 3, and then 3 to launch. So you can sort of break it down. And there are a number of interesting patterns in this. For example, there are a few therapy areas where genetic evidence is actually somewhat differentiating on likelihood of going from a preclinical program to a clinical program, in particular metabolic, which happens to be one of the areas where we feel like we have the best preclinical models for de-risking drug discovery and development programs. So this one's not so surprising to me. This one is surprising to me, neurology. I didn't expect to see that, but those genes with genetic evidence. Maybe there's a little bit of reverse causation going on here by this point in time. I actually didn't keep that part of it more. For the most part, genetic evidence doesn't really differentiate phase 1 to phase 2, as you expect. These are really small studies. You're mostly just trying to figure out, what dose you can give patients without injuring them. But again, metabolic is one of those few areas where it is differentiated, likely because you have, for a lot of metabolic conditions, you have readouts like measuring an LDL cholesterol that can inform efficacy and have some early readouts that you would use in making your go-no-go decision. Most therapy areas, you start to see the influence of genetic evidence in phase 2, and then some residual in phase 3. These are where you would expect to see it because it's essentially differentiating things that actually had clinical benefit versus no clinical benefit. So that's where you would expect to see that relative success start to be more differentiable. We wanted to break down the amount of genetic information by therapy area just as a visual for the paper. And so we actually created this figure on the left first until we plotted it. So this is showing essentially how much genetic evidence in these different therapy areas has accumulated over time. We noticed, and of course, kind of UK Biobank era is what really kind of drives this last big push. But we noticed once we made this, that the order of these was quite reminiscent of what we had seen in the previous figure. So we added this, and we showed that essentially the more this, and this is related really to the results from the two previous figures. The more genetic evidence there is in a particular therapy area or around an indication, the more likely that the successful drugs will have a hit in that genetic evidence versus the unsuccessful drugs. So they are positively correlated, with a few exceptions, the respiratory being an interesting deviation. Respiratory and oncology are the two that are the furthest off of that line. Yep.
So the increase in genetic evidence, does that mean that we get more genes popping up in us? Like you're accumulating more genes?
Yes, yeah, that there's just more for a given indication. So an indication can have lots of different traits that are similar to it. But for a given indication, how many similar traits are there? How many genes have been associated with similar traits?
Yeah, so I think this comes back to the same discussion that I was just having, is that perhaps the idea is that your denominator, now you have so many of them that you're gonna hit. Like if you have a study, it's gonna be targeting one of them. If you were to do GWAS to the entire population of the world, maybe every single gene will pop up as a genetic evidence for any given trait.
But if that happens, RS will go to 1.
I see.
So RS will go to 1 in that instance.
Got it.
In this case, RS is kind of peeling further and further away from 1. So I do agree. I think that, with an infinite sample size, you probably can find some non-zero effect of every gene on every disease. So there's got to be some effect. I used to present a slide on the oligogenic model from Jonathan Pritchard, and talk about core and peripheral genes. Maybe it's that, maybe it's something else. I don't know.
Yeah, maybe waiting with the effect size when you account for when you're calculating the relative success.
Yeah, again, we didn't see any relationship between effect size and relative success. So I'd argue, and we've got lots and lots of nice anecdotes of things of relatively small effect that are really important. HMG-CoA reductase is kind of a key example of that disease.
Did you cluster by gene family to see if perhaps some of this exponential rise was due to like a snowball effect, like you found something in one pathway and then you learned things that were associated in that pathway, and so you studied in that. So you're basically just traversing one branch in a tree and you're just going down to the leaf.
We have not done that, but that's probably worth trying to think of how you would express that in an analysis. We have looked at, for example, protein families. And there are certain protein families that have historically higher success rate. GPCRs is actually the most differentiated protein family in terms of the likelihood of success versus other protein families. Other protein families, not so much, but yeah.
I guess I'm asking in the sense of like people want to study genes that they know are related to other genes that have been successful in the past. So is this kind of like a researcher bias that's also affecting some of these findings?
It's an interesting question. Could be. I don't know that would affect the relative success. That might help explain if you were trying to kind of create an explanation for how we get to different drug mechanisms. I would not at all be surprised if you could find some signal there. I don't think that's influencing the relative success, although they may be correlated. I don't know. Yep. This is another figure from the supplemental material that I think is worth if you're in the field looking at and thinking about. This is similar to that original figure from the 2015 paper, but now broken down by different therapy areas. So this is the probability of genetic evidence, the probability that a target indication pair has genetic evidence based on the furthest stage that target indication pair has progressed across these stages. So this is this is essentially the evidence that's kind of giving us that 2.6 estimate. It's covered up by this. Remove that. No, I can't. But there, although the pattern is similar, that gives rise to relative successes greater than one for all these different therapy areas, the actual probabilities that any of these have genetic evidence are quite different. You know, oncology and infection, for example, they both have relatively high relative successes from phase 1 to launch, but the probability that a launch target indication has any supporting genetic evidence, based on how we define it here, is like 3%. It's a very small fraction. So genetics is differentiating, but you know it's going to have a very low negative predictive value overall on this. Whereas something like metabolic, 50% of the launched target indications are supported by genetic evidence. You know, that's one area that I would not touch a program that did not have supporting genetic evidence.
Sorry.
No, go ahead.
So I'm assuming that in oncology, you're looking at the somatic evidence.
In this figure, we're just looking at germline.
Oh, okay. So, because I was wondering if, because you have a lot more, probably effect sizes in methodologies.
Right.
Compared to if you have somatic mutation that only affects it, you know, small number of patients.
Yeah, driver gene antigen, if we focus just on the driver gene data, you would see a bigger relative success there. Yeah.
So when you talk about the genetic evidence, right? I think you said that like link the variant, because variant said to its presumed gene.
Yes.
Based on linkage equilibrium, that's the main.
I mean, so in this case, for the GWAS, we're mostly using open targets or co-localization. So the open targets bring to gene mapping algorithm, L2G, or EQTL co-localization as the mechanism.
So, I mean, the possibility of kind of long range regulatory effects is not really.
There could be long range regulatory effects for co-localization, but the L2G is heavily weighted by distance from transcription start site. So it is mostly finding the most
I mean, it's
probable.
curious to think about why those drugs that don't have that indication, kind of what are the characteristics of those and why you don't find. I mean, you could imagine a situation where your variant really is, you know, affecting something further away that you just don't cut by that definition.
I'm sure that there are many examples of that in there, but that would not influence relative success because that's just as likely to happen in a genetically supported than an unsupported mechanism.
Something like that. You're able to capture more things that just do not show up because of
Right.
relations.
Yeah, so our probability of genetic evidence could go up.
Yeah.
You know, essentially, we would kind of fill in the void of missing genetic evidence. Interesting time, I'm going to skip that. And this was, you know, another question that often gets asked is, you know, are we already working on everything we should be working on? And this is so way of just saying we've got a ton of genetic information out there and we've only worked on a very small fraction of it. And this is just breaking it down. These are the therapy areas here. This is looking at what fraction of the potential target indication or gene indication combinations have ever been explored in the clinic. It's 2.1% overall. And then this is a heat plot, if you can see it in this contrast here, but it's mostly, you know, there are a few areas that have been pretty highly saturated. It's mostly in kinases with oncology. That's the area that has been most extensively mined of areas with genetic evidence. So the bottom line from all of this, you know, what impact can genetic evidence have on the overall R&D productivity? It's figured from before. If we were to stratify, if we use the clinical probability of success, differentiating those with and without genetic evidence, then our estimate for each approved drug with supporting genetic evidence costs around $650 million per approved drug, and about $2.2 billion for every drug lacking genetic evidence, if you were to stratify on that. So of that 1.2 billion that I previously pointed out, you could if your portfolio is biased against genetics, you're going to be far less productive. If it's biased towards genetics, you can expect to have higher rates of productivity. Again, kind of the Regeneron strategy. All right, I'm going to take a few slides and just talk about using the same type of game to think about other types of evidence. You know, what other kinds of evidence can be differentiating in trying to figure out, okay, I have genetic evidence, now what else should I focus on? This is results from a paper that was published in medRxiv earlier this year from Sarah Takeman collaborating with some scientists from Sanofi. And here they were trying to look at single cell data. They used the Chan-Zuckerberg data set. They identified all of the studies where they had single cell expression data for both cases and controls for 19 different conditions. And they looked at different metrics of differential expression. So at the cell type level, and then at the disease level within cell types, to determine whether that single cell expression data might differentiate probability of success. So they have, you can see down here on the right. So these are their different progression stages with the odds ratios. So it progressed beyond phase 1, progressed beyond phase 2, progressed beyond phase 3. And you can see that, they looked at different subsets, so comparing against all protein coding genes, small molecule tractable, antibody tractable, and known drug targets, similar to what we had done, and showed these really big odds ratios. Well, I read the paper and kind of immediately raised all kinds of red flags around this. So they actually went through this process of using the gene expression patterns for selecting which genes they were going to analyze, and then did not account for that in their analysis. So then they compared it against all genes, regardless of their gene expression patterns. That was really the biggest problem with their analysis. There was a big problem in how they discussed success on top of that. But if you even take the data that they published, and if you identify those things that passed phase 1 in their phase 1 success criteria, conditioned on that, and then ask conditioned on that, what's its likelihood of progressing to phase 3 or to launch, and you actually find that the estimates have odds ratios less than 1. So I mean, bottom line, if you've seen this paper or you've referenced this paper, the results from this paper are not legitimate. I've discussed it with Sarah, and they've redone the analysis and have found that yes, it's not single cell RNA-seq. At least this data in this context was not at all predictive of more successful drug programs. So we can be a bit more rigorous and do this in different ways and look at different types of data to explore this. Yeah, sorry, can't get rid of that. This is asking whether overall gene expression patterns are associated with probability of success. So we're looking at 2 different metrics here. We're looking at the median expression across tissues in GTEx. So if you have a high median expression, that means it's expressed at a reasonably high level across lots of tissues. This other one that we look at here is that it's essentially not expressed in at least 50% of tissues. So 0 TPM and greater than 50% of tissues. So just looking at that median expression across tissues, breaking that down into quartiles, we start with all the preclinical going through. So it kind of tells you, are there biases in terms of types of genes we tend to look at in preclinical? And then you can say, well, once you start looking at them, how likely are they to progress? And this metric actually, so those that have relatively low median expression across tissues are more likely to be successful. This is relative success. And those that have high expression are far less likely to be successful. If we just look at those that have 0 TPM in more than 50% of tissues, suggesting that it's pretty tissue specific in terms of where it's expressed, we see a really clear association pattern. This is not new. This has been found previously in other work.
So
Yep.
this is related to, which I suppose, side effect instead of the causality.
Maybe. Yeah. I don't know. It may be that you're warning against safety effects, and you expect to see that play out throughout clinical development. Different kinds of side effects that emerge at different times. So yeah, I think this is probably a mixed bag of efficacy and side effects, or it may be that your ability to get a drug up to a dose high enough to have the effects you want without causing side effects. Yeah, I'm sure that's playing into it. Another way to look at this is, which is correlated with, but not the same as is kind of some metric of how variable or tissue specific is your expression. And we've looked at a few different metrics. This is an example using Gini index. So this is, where you've got fairly high expression or low expression across lots of different tissues. You have a very low Gini index close to 0, where you have, fairly restricted expression. A high Gini index here at 0.7, 0.6. And so you can look at Gini index and doing the same thing, breaking Gini index down by quartile. You can see that those that have lower variability, that is more even expression across tissues, have a lower relative success than those that have higher Gini index or greater specificity. And then we stratified that based off of the gene expression quartile versus the Gini index quartile. So over here, these are going to be things that are very cell type specific and generally low expression overall. And that's where we see the highest effect.
Are those genes also the ones who are more likely to be hit by patient that shows up in GWAS related to diseases to those tissues?
I don't know. Sure, somebody's looked at that. I don't think I've looked at that.
I guess the colon is right.
I don't know if that's. I'm sure somebody's looked at that. I can't remember the result though.
I'm just wondering if you're looking at the same thing, right? Like
if the genetic evidence is correlated with this.
Yeah.
That's a good question. That would be easy to check. Okay, yeah, we'll check that. I'll just show you one other example, just kind of along these lines. We came up with our own metric of, so a gene can have lots of different transcripts. And GTEx is actually, they use leaf clutter to try and figure out, map all the different transcripts that they think are being expressed in each different tissue. And so just a subset of those GTEx tissues. And you can tell really great from the colors here, but we came up with an estimate of the effective number of transcripts. So you may have a gene that may have 10 reported transcripts that get expressed at some level, but one of them is expressed at 95% of the time, and all the other 9 are only at 5% of the time. So that would, in our metric, that would have a effective transcript number of close to 1. Whereas, here's an example here, where you've got 2 transcripts that are expressed at roughly the same level. Your number of effective transcripts is 1.95. So essentially, it's telling you how many unique transcripts. Here, we were trying to get at the idea of if these transcripts are resulting in slightly different protein products, that our hypothesis is that things that are higher complexity probably have a lower probability of success because the drug, probably is not binding all of them the same. They're probably not all doing the same thing in every cell type. So that was the idea, and we see some evidence to support this. So even after the one on the right is really one to focus on. So here we looked at just those genes that are expressed at relatively low levels across tissues. Here we just did TPM less than 1, and we asked for genes that have more than 1 transcript is the comparator group. So actually, we're looking at low level of expression, only 1 effective transcript, and you can see some evidence that from phase 1 to launch that those genes that have lower transcript complexity are more likely to be successful. So anyway, it's just to suggest that there are a lot of different ways that types of evidence that we can explore when trying to accumulate it and think about what should we work on in clinical development.
Could it also be that in a disease, there's 2 ways a transcript could lead to a disease? One is through expression and one is through alternative splicing. So it might be that that's what's driving the signal. It
could be, yeah. Yeah, I mean, we actually just did this analysis a couple of weeks ago, so we have not plumbed this yet. Yeah, definitely could be lots of different explanations. All right, so this is now a little bit of a sidebar. So in thinking about, we revisited this publication. So when we published, we were essentially using 2014 data. So, there wasn't a lot of genetic information out there. There have been several large GWAS and several things in study, but most of the gene trait evidence has emerged really over the past 10 years since we had done that study, and a lot of it driven by UK Biobank and then some other things as well. This is the data from the GWAS catalog, so kind of just taking all of the top associations from GWAS catalog. This yellow line is the number of unique gene trait associations reported in the catalog. So, we're roughly around 300,000 unique gene trait combinations that are flagged in the GWAS catalog. there's, maybe there's some evidence that is starting to flatten out a little bit, which makes sense because we're kind of have done a lot of the analysis of most of the at scale genetic resources that are out there. There are a few more that are likely coming online over the next few years. Some of the Asian data sets, Singapore among them, are in the 100,000 to 200,000 range. You've got all of us that's, I'm sure there are a lot of groups that are starting to analyze that. It's another 250,000 add on top of biobanks, UK biobanks, 500,000. MVP recently released their summary statistics. They had roughly 50,000 significant gene trait associations that came out of that study. Not all of them novel, but many that were novel. And starting to get into populations that are kind of falling outside of your typical Northeastern European or Northwestern European data sets. But there's nothing kind of in the offing that's going to take us up an order of magnitude in the number of subjects that we can do these genetic analyses in. So in thinking about kind of where are we today, what kinds of genetic discoveries can we expect? I thought about this just from a power perspective. So I've got different trait prevalences here. No, sorry, these are different minor allele frequencies for the variance. Prevalence in this one is across the x-axis. So, and this is on a log scale. So you have prevalence of 1 in 1,000 up to 3 in 10 on the right-hand side. Genetic effect sizes are the different lines here. So starting at 1.025, which is kind of where we're finding things in the big, hypertension type GWAS, all the way up to the ratios of 2 and 5, which are only big effects that we are finding in some of the loss of function studies that we're doing. And so you can see that, as you would expect, for genetic variants that are relatively common, for traits that are relatively common, we've probably found most of the associations that are out there to be found. But even for common variants, once you get it down to traits that have lower and lower prevalence, this is 1%, this is 0.5%. That's not that rare. Most autoimmune conditions fall somewhere between 1 in 1,000 and 1 in 100. Only a few of them are up in the 2.5% range. So even the things that we commonly talk about, we're really, even for really common variants, we're not that well powered to have identified them. So I think, there's still, as we increase our sample size for GWAS, we have, there's still this kind of low frequency but not rare condition range that I think we still have a lot to discover. But as soon as you start thinking about things that have relatively low frequency and relatively low penetrance, we have almost no information about them until you get into the Mendelian side of the effect size scale. And people think, oh, well, we can do these UK biobank type aggregate analyses of loss of function, and that's going to kind of recover a lot of our power. This is actually an analysis of NOMAD across the different subpopulations highlighted in NOMAD, looking at the cumulative allele frequency of putative loss of function variants in NOMAD. So this is that distribution of that across the 18,000 or so genes that that we have the NOMAD data for. And the overall, it's hidden there, but the median cumulative minor allele frequency of those PLOFs is 0.02%. So kind of going back to my power slide here, it's below the top, the frequency of the top figure that I have there. So even trying to play this game of looking at these, potentially highly impactful loss of functions, we're completely underpowered, even for fairly common traits. Once you get outside of quantitative traits that are very biologically, genetically strongly determined, we have very little power really to study most of this. So what we have today is really just skimming the top. And so I was trying to relate this back to the same figures before, but I prepared this talking to the BioView Consortium, kind of what they could expect to find from having whole genome sequence the 250 to 300,000 people in the Vanderbilt's biobank versus, maybe over the next 3 or 4 years, you can aggregate a data set of 3 million of these. But even with 3 million of those, if most of your genes, more than 50% of your genes have frequencies less than this, again, it's only the most common conditions that we're going to have reasonable. So this is an odds ratio of 1.2, for something that's kind of a modest effect. Here's an odds ratio of 2. So yeah, pretty big effect size. So even with 3 million subjects in that type of loss of function analysis, it's only the pretty common traits that you're going to have any reasonable power. So kind of my point in going through all of this is just to highlight, this is kind of a busy slide, but this is my quasi-informed guess at how much of the underlying genetic evidence that exists in the human population that we have discovered to date. And so I've got minor allele frequency on the x-axis here, the percent of potential genetic evidence that we've actually discovered. So, for Mendelian stuff, we've discovered most of it. For common frequent stuff, we've discovered most of it. But really for the rest of this, we barely scratched the surface. I think we may know, 1% of the same types of genetic effects that excite us as human geneticists today. This is just another way to look at it and think about it. This is from a weird paper in the Journal of Pathology from 23andMe. They've got great data sets for doing this type of analysis. They looked at how did effective sample size in thousands affect the number of associations, the number of significant loci that were discovered. And I actually have an overlay that I added onto this. So here's each one of these lines is a different trait that they did this analysis for. So there's some traits that, as you increase your sample size linearly, you increase your number of discoveries linearly. But actually most traits kind of falls below a 1 in 10. So you need a 10-fold increase in sample size to double your number of genetic associations that you can identify there. So anyway, that's that for that digression. And I'll just finish. I know we're at time. We've got a preprint out there on genetic associations and playing the same type of game on side effects. And this is with side effects for approved drugs. And bottom line, we show similar types of things that and not surprisingly, that side effects that are relatively common for approved drugs, they're more likely to have a genetic association, indicating that there's a relationship between the drug target and the side effect in question. Probably most impactful in heart-related conditions and oncology, I would say. So in the bottom line is, genetics has a lot to offer in the drug discovery process, and I think that it's going to be an important part of it for quite some time.
So Kay in the chat says, hi, it seems like you've identified a large space for future drug discovery. Do you have a specific approach to identify a specific drug for Genscience to develop? Or is your company more interested in surveying the space generally?
Yeah, so we will not become a drug discovery company because, one thing I've discovered in 5 years at an investment firm is it's almost impossible to be a good platform and to be a good drug discovery company. All companies that started out with, most companies that start out with a good platform idea that try to then do drug discovery, kill the platform. Because once you have that first program going into phase 1 and requires all the capital, the capital for anything else dries up because your investors, all they're thinking about is what is their return going to be? And that return is not going to come from the platform. It's going to come from that drug in development. So yeah, our focus is really around trying to build that best gene disease hypothesis and how to think about using that for making good drug discovery decisions.
And then Cindy, you want to unmute yourself?
Hi, thank you for the presentation. I think it might be a relatively minor question, but I noticed there is a therapy area called sign and symptoms when you're talking about the value of genetic evidence. I'm wondering what exactly it is. Is it just non-specific symptomatic treatments that doesn't belong to any of the categories?
Yes, it can overlap with other categories. So those categories are not all one-to-one. So a single trait can map to multiple of those. So you can just think of it as here we were using mesh as our ontology. We've now transitioned over to Mondo as our primary ontology, but in mesh, a trait can map into multiple different branches. And so and each of the major diseases, those therapy areas have a branch. And it's just indicating that that trait maps into that branch, but it could also map into another branch. And so in that case, those data were reused multiple times in that analysis. But the signs and symptoms is just kind of a catch-all for, so something like, I think, systolic blood pressure maps into signs and symptoms. So those types of measurements.
Okay, thank you.
We are at time. I'm not sure if you have a hard stop or if there's any.
I don't have a hard stop, but anyone who needs to get up and leave, feel free to leave. If anybody wants to stick around and ask any other questions, I'm happy to stick around.
Any more questions on Zoom or in the chat or in person?
I have a question. So you mentioned, I think, at some point that you guys started to see if you can make the contribution of the variance based in some meaningful ways. For example, using like the effect side. Another possibility is to look at networks. And try to see what is the position of the gene in the patient network and how that maybe make that. Have you guys thought about it or looked at this at all?
Yeah, I really like that idea. There have been some good preliminary studies that have examined that. So the GSK group did that using HotNet 2. And Open Targets published a paper where they used a PageRank algorithm for doing that, where they built networks using different types of data. I think for the Open Targets paper, they used, I think they just used Reactome, maybe Reactome with StringDB. And I think the GSK paper used Metacore. I think the Metacore pathways for doing that. But essentially, you take however you get your network or your pathway, you overlay the genetic information, and then you use some sort of diffusion algorithm to then say, what is the heat, in the various locations of the network. And that actually works pretty well. Yeah. So I mean, it seems to work similarly to having direct genetic evidence. So if you've got a target without genetic evidence sitting here, but it's in a network in a neighborhood that has lots of genetic evidence around it, propagating that genetic evidence to that seems to be almost as informative as having direct genetic evidence.
Just Richard, his ideas are somewhere over the line like that. Confused like that.
So I was struck by the lack of association between effect size and the use of the information process. But I'm wondering whether there's actually, for example, if we take the odds ratio, is there qualitative difference between 1.05 versus 1.001? I think I don't know if you look into the data in since a lot of the newer biobank data may actually switch on various peptides. It is, I just wonder whether it's actually, if you think go back to Richard's core genes versus preferred genes, I would guess there should be a difference in terms of how useful or how
I just don't think effect size gets at core versus peripheral. I think that's the issue. I think if you could differentiate core from peripheral, I would anticipate that would be associated with relative.
But that was, yes. So we cannot assume, but I wonder if it's actually the range. But if you think about 1.05, 1.1, they're basically they're all in the core gene. But further down, it's really like we typical in infinite or like infinite or that's like there's thousands and thousands of those that contribute to height. I think that's these cannot be the core. Well, at least that cannot be directly involved in the biological process.
Yeah, I think if you were to take some of these really high-powered GWAS, like type 2 diabetes now, and some of these, and so let's say if you had 20 traits that had over 1,000 associations, I feel like you could probably start to, and if you stratified by frequency and effect size, so if you can somehow control for your lower power to find the rare higher effect size things. It feels to me like you should be able to start to tease out peripheral from core if that's, I mean, I think that's a reasonable model to think about this, but yeah, we just haven't figured out a way to do that yet. I think there will be an effect there if we can.
Yeah.
But I don't think effect size itself, effect size by itself does not tell if it's a core or peripheral.
Yeah, change.